Hey there, in this video we are going to be reviewing the process of cell division known as mitosis. So this is the mitosis activity. You are going to need the mitosis template, and you are also going to need one of these small baggies with the different color of pipe cleaners and the beads for this activity. So make sure you're using this sheet as a layout template. You will first place your model chromosomes in each cell, circle according to the rules and assumptions listed on the procedure sheet. I am going to be your procedure sheet, or you can ask me for a paper copy of it, but as long as you are following along in this video, you should be able to complete this activity. And of course, if you have any questions or concerns as you are completing this activity, remember you can always pause, raise your hand, and ask a teacher in the room if you need further explanation. So we're going to start off in our first cell. This cell is technically right before prophase would occur. So we are going to start with 2 chromosomes. Now remember, chromosomes are simply packaged DNA, and that package is held together by our centromere. So the beads are going to represent our centromeres here for this activity. So before any type of cell division can occur, cell division like mitosis, our chromosomes need to go through DNA replication. So we are going to simulate DNA replication by adding in a second pipe cleaner that is the same color as our first one. So I go from having 2 single chromosomes to having 2 double chromosomes. So here is my first cell in prophase where I have 2 double chromosomes. So they have been replicated. So in prophase, our nuclear membrane begins to break down, and then we head to metaphase. So I'm going to grab 2 more pipe cleaners and my bead for my centromere. So in metaphase, my chromosomes are going to line up in the middle of the cell. So metaphase middle. So that looks like this. There is no rhyme or reason. There's no rule that tells chromosomes how they need to line up. They just simply line up in the middle. Again, this is a model of mitosis. If we were looking at actual mitosis within human cells, we would have 46 chromosomes stretched from one end to the next. We don't have enough time or materials to do that, so we're just representing mitosis with 2 chromosomes instead of 46. But do keep that in mind. There's no rhyme or reason to how our chromosomes line up. So I could have put the purple one up top if I really felt like it. Okay. The big thing is that metaphase, we line up in the middle. Then in anaphase, so I'm going to get 2 more chromosomes. In anaphase, our chromosomes are pulled away from each other. Oops, sorry. I messed that up a little bit right there. Here we go. So this one and this one. There we go. This one and this one. There we go. So I left the centromere on one of them just as a kind of a visual reminder that yes, they are technically still there because the DNA has been packaged up. So in anaphase, our double chromosome gets ripped apart. Right, and each sister chromatid, right, is sent to the opposite end of the cell. Then in telophase and cytokinesis, right, we now have 2 daughter cells, each with a copy of the original chromosomes. So we started with 2 chromosomes and we ended with 2 chromosomes. So the daughter cells of mitosis are genetically identical to the parent cell at the beginning of mitosis. Again, only a slight difference, right? We had to replicate our DNA. We had to replicate our chromosomes. But that was to ensure that both daughter cells have the correct amount. So in mitosis, we do not gain or lose any DNA. The daughter cells are genetically identical to the parent cell. Remember, mitosis serves two main functions. We need to go through mitosis. We need to make new cells because some cells can become damaged. Right, you get a paper cut, right, you damage those skin cells, and so they need to be replaced. We also have cells go through mitosis in order to grow, right? You physically grew taller from last year, and that was because you now have more cells within your body. So growth and repair, the two big reasons why cells have to divide. And again, these are body cells like skin, lung, liver, spleen. So body cells go through mitosis in order to repair damaged cells or for the organism to grow. As you see, I did not use all of my materials. That's perfectly fine. But I did use enough material to represent the DNA for the entire series for every stage of mitosis. Okay? So if you have any other questions or concerns, remember our big takeaway here, the number of chromosomes we started with in the parent cell is the same as the number of chromosomes in our daughter cells. Okay, so hopefully this kind of helped to give you a visual representation of what happens during mitosis. If you still have any questions or concerns, just let me know.